Welcome. In this lab session, we will learn how could we predict and model the 3D structure of protein and how could we analyze the tertiary structure of protein. So in this lab session, we will learn by using the Phi2 tool, how could you analyze or predict the secondary structure and how could you model the 3D structure of protein. So let's now understand how the Phi2 tool is used to predict all of these information. Phi2 tool is a suite of tools that is available on the web to predict and analyze the protein structure, functions, and mutations. The focus of Phi2 is to provide a very user-friendly interface to the scientist or biologist. Phi2 uses the homology modeling algorithm to build the 3D models. It can also use to predict the ligand binding sites and analyze the effect of amino acid variants. And this tool is freely available for the non-commercial users. So let's now understand how can we use the Phi2 for the prediction of secondary and the tertiary structures of your query sequence. To get into the website, you just have to click on the link mentioned above, or you just have to write the Phi2 on the Google, then you will be redirected to their homepage. This is the homepage of Phi2. You just have to click or you just have to paste your query sequence into this section, into the amino acid sequence section. And then you just have to click on the file search. This tool will take some time to generate all the results. As an option, you can give your email address over here so that you can get the link of your result on your email ID as well. So after few hours, depend on the length of your query sequence, you will get the results. You can download all of your results in a zip file as well. As you can see over here, first of all, we have an option of a summary. In the summary section, there is a 3D model over there, the image colored by N terminus to the C terminus. This 3D image has been modeled with a confidence score of 99.5% and with a query coverage of 25%. So now the question is, how could the Phi2 tool use? When you paste your query sequence, first of all, the query sequence is scanned against the database SciBlast for the multiple sequence alignment. And this multiple sequence alignment is then used to predict the secondary structure database. Like you just have to click on the multiple sequence alignment, then you will get the information about the multiple sequence alignment. This information is used to predict the secondary structure of your query sequence. As you can see over there that this is your query sequence. The green color represents the helix, the blue arrow represents the strands. Just like that, in the alpha helix, which is over 31% for this query sequence, is represented with the helixes in the green color, with the coils in the green color, you can say, with the helix shape. And then the beta strands is only of a 6% that is represented with a blue arrow. And there is another subtype of helix that is a TM helix that has been represented with a brown color helix or a golden color helix, whatever it represents, and that is of about the 3%. And you can also see you can also view the membrane topology prediction if you want. If one would like to get the information regarding the domain, then you just have to click on the domain analysis, then you will be redirected to this result. So these are the information regarding the domain of your query sequence. You can also get the detailed information about the templates that has been used for the modeling of the 3D structure. So just you have to click on the detailed template information, and then you will be redirected to this page. As you can see over there that we have a 3D models along with their confidence score and percentage identity and the template information. So on the basis of this information, you can select your desired model. But according to that, the model number one is with a high confidence score of about 99.5% with a percentage identity of 100%. So you can you can download your desired model just by clicking on this model, and then you can save this model in a 3D in your PDB format. If one is interested for the mutation analysis, then one can find the quality of their models and other functional side. Then you just have to click on Run Investigator. So in this way, by using a single tool, you will be able to predict the secondary structure of your query sequence. You can also get the information regarding the domains, about the functional sites and mutations, and more importantly, you can easily model your 3D structure of your query sequence by using the homology modeling algorithm. So what you gonna do is you just have to put your query sequence, just paste on the query box, and then just click on the file search, and then you will be redirected to the result page after a few hours, and then you can use all of this information accordingly in your research. Thank you.